From Microbe TV, this is Matters Microbial, a podcast about the wonders of microbiology, microbiologists, and microbial centrism. This episode was recorded on January 16th, 2025. Hello, Micronauts, and welcome to our Quality Quorum for today. I'm your host, Dr. Mark Martin, Associate Professor of Biology at the University of Puget Sound in Tacoma, Washington. Today is the 75th episode of Matters Microbial. Can you believe it? Thank you for every view and every listen, and for spreading the good microbial word. As anyone who has ever met me understands, I have a lot of microbially themed enamel pins. But I recently saw one I do not have, seen here. And I do talk constantly about microbiology. If I find a vendor, I will share it here. As I move through phased retirement, I no longer teach introductory cell and molecular biology, sadly. Anyway, one topic I enjoy is helping students to understand the relationship between structure and function, which I call stericity, a wholly made-up word. Consider starch, one of the subjects for today, seen here. It is the way that living things can store up food or energy by polymerizing glucose. The links between those glucose monomers are important to functionality. Cellulose is also made of a polymer of glucose, seen here, but the bonds between those monomers are ever so slightly different, and thus the functionality is different. Comparing the two polymers, seen here, is a great student exercise. French fries are made of starch. Pencils are made of cellulose. But they're both made of a polymer of glucose. The differences in innate structure are actually subtle and small. But these differences are important. Only bacteria can break the bonds between the glucose monomers and cellulose. So, termites don't break down cellulose. The microbes in their gut do. But starch is easier. Our very saliva contains the enzyme that can release those glucose monomers called amylase. Here's a great exercise for students. I make plates with starch. Starch stains dark, almost black, with Lugol's iodine solution. Glucose does not stain. So I had students wet a swab with their own saliva, draw on the plate, wait 20 minutes, and stain with Lugol's iodine. Have a look. As you can see, wherever the saliva touched, broke down the starch in the plate. We eat many different types of polysaccharides, including starch. How do gut microbes deal with them? For this reason, I am so very happy to welcome Dr. Darrell Cockburn, Associate Professor of Food Science at Penn State, to the podcast. Welcome to our Quality Quorum today, Darrell.
Hi, Mark. Thanks for having me.
Oh, this is such fun. And I mean, it does take me back, as I say, to intro biology. I hope you weren't cringing too much at my description of things.
No, that was great. I like hearing about both starch and cellulose because I've worked on both of them in my career. So.
And I'm well aware, and I'm sure I'm going to have folks write to me or on YouTube point out that I didn't mention branching, and I didn't mention, the hydrogen bonds between the fibrils and cellulose, and that's true, I didn't. But I was sticking with the higher level of stuff. So I think a good place to start is why we should care about what type of, I should say first, let me do this a different way. Routinely, I get people talking about carbs, and I cringe every time. Because one thing that I teach my students in intro bio is that the term carbohydrate is way too generic. And so for that reason, I think the focus should be on polysaccharides primarily.
Yeah, no, I agree. There's also, various mono and disaccharides that are important in our diet too. But my research is really this focus on polysaccharides and the dietary polysaccharides particularly. And so, why should we care about those? They are a major source of our calories that we get in our diet, but they are also a critical energy source for the microbes in our gut. So you can have those carbohydrates in our diet, you can divide those up into categories in a number of different ways. So we often will talk about starch and fiber, but there's also a lot of subdivisions within those categories. And it determines some of the sort of health impacts of those carbohydrates in our diet. So if you take starch, for instance, you can divide starch kind of into three different categories of rapidly digestible starch, slowly digestible starch, or resistant starch. And so the rapidly digestible starch, that's going to be what we call high glycemic. So it's going to behave almost like you were just eating sugar. So if you look at what happens to your blood glucose levels after you, say, eat a slice of white bread versus just take a glucose solution, they're very similar. Because our body is really good at breaking down some of that starch. With that salivary amylase gets it going right away as soon as it enters our mouth. If you keep that piece of white bread in your mouth and let it sit for a little bit, it'll start tasting a little sweet over time because you're releasing those sugars out of there that you can now sense.
That's, by the way, how I first learned about this as an undergrad. Back when dinosaurs ruled the planet, we were supposed to bring some white bread in, and we kind of mushed it up to make it, and then we tasted it. It tastes like starch, and then you would put it down, and then after 10 minutes, put it back in our mouth. Everyone gets grossed out when I say that, but trust me, people eat a lot worse daily, and it tastes physically sweet. I can't do that with students, but it's exactly what you said.
Yeah, so that's your rapidly digestible starch there. But we don't just make amylase in our saliva. We also have a pancreatic form of it that gets secreted into our small intestine. And so the starch goes through our stomach, and the acid in our stomach is not doing a whole lot to the starch. It might help start breaking down some of the physical structure a bit more. And then we have these pancreatic amylase that will start breaking down further. So we have this second category of starch, slowly digestible starch, and that takes up to a couple of hours to get broken down by our amylases, but we still totally break it down and are able to access all those carbohydrates. But it doesn't lead to the same sort of blood sugar spike because it's over a longer period of time that you get that broken down. But then we have this third category, which is resistant starch. And that's the starch that will survive all the way through our small intestine and make it to our colon. And now it becomes more like dietary fiber and is an energy source for our gut bacteria. And so there can be a lot of reasons why that starch has different rates of digestion. So the easiest one to understand why we would get it to being rapidly digestible is because we cook it. And so this breaks down all the structure of the starch. So if you look at when the plants are making it, they make it in these nice starch granules, which are these sort of semi-crystalline large constructs. And the plants need to use those as an energy source, in their seeds, for instance, to make a new plant. That's the energy source that it's using. But they break that down over the course of days, weeks, as they're accessing that. So they don't need this super rapid access to that starch. But we need to digest it much more quickly than that. So we tend to cook our food, and that makes it much more quickly digestible for us. And so what that does is it makes it, that starch undergoes a process called gelatinization, where essentially the combination of heat and water breaks down that granular structure and kind of separates it out into the individual strands of the polysaccharides that our enzymes are really good at breaking down. So then, how do we get these other forms of starch? Well, if you take, your cornstarch, and if you just eat that cornstarch raw, that's going to behave more like a slowly digestible starch. So these are going to be the starches that are not fully gelatinized, and so our enzymes have a harder time breaking them down. They still can over the course of hours, but it's a slower process. So if you have an incomplete gelatinization or no gelatinization at all, then it takes a little longer. But then where does that third category, resistant starch, comes in. So we have 5 different categories or types of resistant starch. So the first one is the whole grains. When you have all this material surrounding the starch, so you're physically blocking access to the starch by the enzymes, that allows the starch to survive passage through the small intestine and make it to the colon. We can also have type 2 resistant starch. So this is a little bit trickier because this is different plants making their starch in a little bit different ways. So if you look at different starch granules under a microscope, you take cornstarch granules and you take potato starch granules and you take starch granules from all kinds of different plants, they don't all look the same, even though they have that same molecular structure that you mentioned of these glucose monosaccharides being linked together either through alpha-1,4 linkages or alpha-1,6 linkages to form branches in the starch molecules. But it turns out that the way that you make it in terms of the lengths of those chains or the degree of branching that you put in or where those branches are can end up changing the properties of those starch granules, including how easily digestible they are by enzymes. And so potato starch granules are not as easily digested by our amylases. So wheat and corn, they make what are called A-type granules, and tubers like potatoes make B-type granules. And I won't go into all the molecular details of exactly how they're different, but it turns out that if you take, so I said if you took raw cornstarch and ate that, that would be a slowly digestible starch. But if you take raw potato starch, that will be a resistant starch. Because our amylases don't properly recognize the surfaces of those and don't as easily penetrate into them to break down the starch molecules there. Now, if you cook your potato starch, you still are going to end up breaking it down to that rapidly digestible starch. But it's going to depend a bit on how you are cooking, how long you're cooking, how much water is present in there, because that gelatinization process takes a combination of water and heat. And so, if you take a big potato, the center of that potato might not get quite as hot as the surface. There might not be as much penetration of water in there. And so some of those starch granules can survive in there. So even when you cook your potatoes, some of that resistant starch can survive. But then, say you make your potatoes, you make some mashed potatoes, or say you cook a pasta, and now you have your leftovers, you put it in the refrigerator. What happens over time is that starch was gelatinized, you've broken down its crystalline structure, but now it can start recrystallizing over time. And this new crystallization is not necessarily replicating what the plant had originally done. It's going to go into its sort of lowest energy state, and it turns out that this ends up also being a resistant starch. So your leftovers are likely higher in resistant starch than your fresh food that you cooked up originally. And then we can have type 4 resistant starch, which is chemically cross-linked resistant starches. So when you look on your food labels and you see modified starches, some of those could be chemically cross-linked resistant starches. And then we can have type 5, where if you look at the individual molecules that make up starch, we've got amylose and amylopectin. And so amylose is just a linear chain, so it's only got those alpha-1,4 linkages. And then the amylopectin has branches coming off of it with alpha-1,6 linkages. So particularly in the amylose, if you look at it, that long linear alpha-1,4 leads to sort of a helical structure of the polysaccharide. And it turns out that certain molecules can get inserted into that helix, particularly lipids. And that can render that amylose and the starch that it's a part of resistant to digestion as well. And people do things with trying to put other different interesting types of guest molecules inside there. It's related in concept to, if you know what Febreze is. So it's a deodorizing spray that people use. And the active thing in there is called beta-cyclodextrin, or other cyclodextrins can be used. And that's essentially a little piece of starch that's formed a circle, and it can capture molecules inside of it. And so that's why it can take the odor molecules out of the air as they get trapped inside that ring structure. Well, the same sort of thing can happen with those amylose molecules. But in this case, because they're a lot bigger than those cyclodextrins, there can be larger molecules that are getting incorporated inside. And that can end up being a form of resistant starch, and potentially also be something that could deliver other molecules to the large intestine. So you could incorporate things in that you want to make it to the large intestine as well.
Is this why iodine tends to stain the starch but not glucose? Is this principle?
Yeah, exactly. So it can incorporate into those helices, gets trapped in there, and so then once you break the starch down to the pieces that are small enough, you don't even have to go all the way down to glucose. Once you make them small enough that they can't form those helices, now it's not going to bind the iodine anymore.
Cool. That's what I'd heard, actually. For listeners and viewers, I mean, actually, oh, there, I said it. Carbohydrate, with generic term, carbohydrate chemistry is super complicated, but it's really important too. If you're just listening to what's going on, I mean, you can hear about the complexities. I mean, I will never look at my dinner plate quite the same way. I'm going to actually have like a cheat card I'm going to make next time I go out for this reason. I was really taken by the observation you made that after you cook something and then refrigerate it, that sometimes the starch-like polymers go back to a less accessible form. That fascinated me, and this sounds like a flippant question, but I think it's relevant. Does that mean that fresh pasta is less good for you than stuff that's been in the fridge for a while, or, Lord forbid, canned spaghetti?
So I think I'll make a bit of a distinction between those two possibilities there. So if you made your fresh pasta, cooked it up, you have maybe even a whole homemade tomato sauce. There's going to be all kinds of nice nutrients in there that are going to have benefits too. What's going to happen to the starch when you refrigerate it is that you're going to get an increase in the resistant starch. And that is probably beneficial. Some of the other properties might degrade over time, that you might lose some other health benefits as you're gaining these ones. For the pasta in a can, they're kind of, now I'm not 100% certain exactly what's going into all the manufacturing there. However, people making starchy processed foods do think about this retrogradation process and actually try to inhibit it. And so I'll come back to go to another example, which is bread, which that retrogradation process happens in bread as well. And that's a major reason why your bread goes stale over time. But when you go to, modern bread products, you see that they're kind of lasting longer and longer and longer in your pantry without going stale. What's happening there? Well, they're doing different types of treatments to inhibit that starch retrogradation from taking place. And I'm not 100% certain if similar things would be happening with like canned pasta, but I wouldn't be surprised if that were the case. And so you might not actually be getting that resistant starch formation happening in those cases.
I think it's good to point that out. I have a former student who works in food sciences, and there are a lot of actual thought that goes into this business, and I'm not being difficult or saying things that are insulting to anyone. They really do think about what would make things better as well as things that taste better. So that it's not an evil corporate thing. It's a matter of making sure that there's good value for some people.
No, exactly. They don't want to make your bread less healthy. They want your bread to last longer on the shelf.
Yes. At least we're no longer using radiation to do that. That
Right.
was a lot of fun in the 1950s. I thought it was really interesting, the business with potatoes. I like French fries, and I know I can only have them occasionally. But you mentioned that there were positive benefits in the way that you actually deal with potato starch when you cook it in the presence of water. This is not taking place with delicious French fries. So do you have thoughts on that? Are we taking away any positive benefit this way?
So, interestingly, if you make French fries or other things where you're cooking them in oil, yes, you will preserve more of that resistant starch in there. It's actually true. And so probably I've never actually done the test with like a McDonald's French fry or something like that. But I know of people who have looked at French fries under the microscope looking for starch granules and can see them actually intact in there at a fairly high rate. So you are actually maintaining them. Now, whether that outweighs all the fat and salt and things that you're also adding on to those things, probably not. However, from a purely resistant starch point of view, you are getting more in those cases.
We are not dispensing medical advice on this podcast.
No.
We are dispensing microbiological advice, however.
Right.
Speaking of that, many times even I get confused at the things that I see on labels for particular foods, especially if you're trying to look for things that we perceive as healthier. What sorts of things should people look for, do you think?
Yeah, so you'll see, sometimes you will see glucose on there, but dextrose, for instance, means the exact same thing as glucose. Fructose, it's just another monosaccharide that we use pretty much just as well as glucose. So high fructose corn syrup, for instance, will be high in that. But also the other part, when it's high fructose corn syrup and not just totally fructose corn syrup, the other part is glucose. And so there's maybe hidden sources of glucose in there behind some of those names. It can be hard to parse apart. The nice thing about the nutrition labels, though, is they do have to tell you how much of the carbohydrate content is sugar and how much is added sugar into it. So those are things to really pay attention to. Probably easier than looking for specific words in the ingredient list, which they can kind of hide and do some things to maybe mislead a little bit sometimes.
What I wanted to say here, and I know you're going to get here, but I want people to know this is coming, is we tend to look at what we do, what we eat, and then we don't think about the microbes that are our partners, but are actually using what we eat. And so many years ago, I had a colleague, a mentor, Abigail Salyers, who looked at gut microbiome issues in the early days, specifically Bacteroides. And this issue of how an organism could eat such a polymer was fascinating to her. And she used to say, how do you eat something bigger than your own head? And I know you're going to come to this a little bit. And I love the idea of how the microbes encounter these things. But so far, we've had a really great description of what goes on with the types of polysaccharides that we're consuming. And I presume that the food industry is, as you mentioned, trying to make modifications, hopefully to level out the glucose bursts in our bloodstream.
Yeah, so that's one application of it, is that if you replace some of the essentially rapidly digestible starch in a food with resistant starch, one of the effects is going to be to lower that glycemic response from that food. So that's an effect that's going to be totally independent of our microbes. That's going to be, directly acting with our own enzymes. But then there's these other sets of effects that are more targeted to our gut microbes. So all these different types of resistant starch, what they have in common is that they evade digestion by our enzymes and make it to the colon. So then what happens once they get there? So there's microbes in our gut that are starch degrading organisms. It's one of the more common functionalities actually that you'll find within gut microbes. However, just like this resistant starch is resistant to our enzymes, it's resistant to a lot of the enzymes of the microbes as well. There's actually only certain specialized microbes within our gut that are able to actually digest that resistant starch and make it more accessible to, say, some of the other starch-degrading organisms in the gut through what we call cross-feeding interactions. So the species that are known to actually be able to degrade resistant starches are things like Bifidobacterium adolescentis and a few other closely related Bifidobacterium species, and then Ruminococcus bromii and a couple of other closely related species. And then one that we've been studying more recently, Clostridium butyricum, which is present in the human gut, but not as quite as high levels as those other two. And so most people will have one or more of those organisms within their gut microbiome, though not everyone. There's actually some people who don't have any microbes that digest resistant starch, and now that resistant starch will act just like cellulose and kind of pass right through their intestinal tract. Because we actually do have bacteria in our gut that degrade cellulose as well, but it just takes too long. Cellulose is a very recalcitrant polysaccharide, as we say, and so it's difficult to break it down. And so over the time scales of things passing through our gut, it goes through. And so similarly, when I say that we have these other bacteria and the resistant starch is resistant to their enzymes too, they can break it down, but again, it's much more slowly than these specialist organisms are able to do it. And so the vast majority of it would pass through if we didn't have these highly specialized organisms like Bifidobacterium adolescentis, Ruminococcus bromii. And so one of the interesting things that we like to study is what sets those bacteria apart from all these other sort of regular starch degrading bacteria in the gut. And so one thing is that their complement of enzymes that they have is a lot larger. And so if you look at us, we have our salivary amylase, we have our pancreatic amylase, we also have a enzyme in our intestinal brush border for breaking down maltose, which is a digestion product from the starch, into the individual glucose and absorb it. But that's pretty much about it for what we've got. Some of these microbes have a lot more. So Ruminococcus bromii is particularly interesting because it forms these multi-enzyme complexes. And so it's analogous actually to how some cellulose-degrading organisms are able to accomplish this. So there's these structures called cellulosomes, where they essentially have these adapter proteins that allow multiple enzymes to join together into large complexes. And so there's a similar thing that exists for starch called the amylosome that's produced by Ruminococcus bromii. And so it's able to bring together several different amylases, several different poolinases. So this is the other key class of enzymes. And these are the ones that will debranch the starch. So they break the branch linkages, the alpha-1,6 linkages. And so it brings those together in combination with another important part when you're dealing with resistant starch, which is binding functionalities. So carbohydrate binding modules. So that's one of the things that our enzymes lack. So our human enzymes for starch digestion, don't have these carbohydrate binding modules, which are separate domains that are accessory domains on the enzymes that allow them to more efficiently target these complex insoluble substrates. And so that's part of the reason why our enzymes struggle to degrade these substrates, is they just don't have the proper targeting functions to concentrate the enzymes on the surface of these intact granules or otherwise insoluble particles and efficiently digest them, whereas these microbial enzymes do. And so they have quite a variety of different amylases and poolinases and binding modules that all kind of come together to facilitate this breakdown. So we're not entirely sure at this point why they need more than one different carbohydrate binding module, or more than one different amylase, or more than one different gluconase. But they do. They have multiple of each of these. And they do have, if you take them and study the enzymes in isolation, they have slightly different properties in terms of substrate preferences, in terms of rates of action, in terms of like what they bind to and with what efficiency. So a lot of our work is we try to untangle all of those relationships to understand what actually makes them capable of doing this. For instance, can we bring it down to a minimal set that we could transfer to other organisms to make novel probiotics or something like that could now digest resistant starch. So maybe I'll now bring it back to why do we care about whether they're able to break this down or not? And this is true of other fibers in the gut as well, is that we want our microbes to be breaking down fiber and producing things that can help benefit us. So they improve our health. And so one of the big things that we focus on in my lab, and is a key thing associated with fiber digestion, is the production of short-chain fatty acids, particularly butyrate. And so these are, we're in an anaerobic environment in the gut. They are trying to break down these carbohydrates, but they can't use respiration. So they need some other way of getting energy out of these things. And so they use fermentation. And within the gut, carbohydrate fermentation usually passes through these short-chain fatty acids as end products. And that's a good thing for us because we have co-evolved with this microbial population, and we have all kinds of sensors for these different short-chain fatty acids. We actually directly utilize them as a calorie source. So our intestinal cells will absorb these. And in fact, butyrate is the preferred energy source of our colon cells. It would rather use that than glucose. And so when our colon cells are growing in the presence of butyrate, that's kind of when they're at their optimal health state. And so you'll see that having high butyrate levels has anti-cancer effects, has anti-inflammatory effects, has pro-satiety effects too. So there's connections through to our satiety hormones in detecting these. So it's kind of like our body is sensing whether we are having a good amount of this fermentation taking place, particularly later in our intestinal tract, as an indication that we're getting adequate nutrients coming in overall. And I think one of the problems with the evolution of the Western diet is we've divorced this relationship between the amount of fiber in the food and the amount of calories in the food. And so now we kind of have messed up this system a little bit. And so we're not properly detecting how much food we're actually eating because we're not seeing as much of this butyrate and short-chain fatty acid production for a given amount of food that we're eating. And so our body doesn't recognize that we have taken in enough. So that recognizing that satiety is one aspect of it. Its anti-inflammatory effects are another. So there's a lot of these interactions with the immune system. And so because we don't get enough fiber in our diet, it means that we are not producing as much of these short-chain fatty acids and butyrate as we probably should. And so we're not getting as much of the signaling happening as would be optimal for our health. So we'd like to be able to increase that. The easy answer is everyone should just eat more fiber. But that's been a recommendation for quite some time and hasn't seemed to be an effective message out there. So one of the things that we look at then is, well, can we understand a little bit more about what's happening with those microbes and see if we can maybe have more targeted approaches so that we can get by with less fiber to get the same sort of net benefits happening. And so to do that, you have to understand in more detail the interactions between the particular fibers and your gut microbes. So with the resistant starch, we're kind of, that's our favorite model to look at. We have all these different types of resistant starches. Do they all behave the same in terms of their butyrate production? It depends on the gut microbes. So if you look at the different, one of the interesting things is if you look at people that have both Bifidobacterium adolescentis and Rhuminococcus bromii, and you feed them different types of resistant starches, you can see that there's a bit of a competition that then happens between those two resistant starch degraders. And the Bifidobacterium adolescentis is really good at breaking down potato starch. So if potato starch is your source of resistant starch, and you have both of those in there, you're going to favor the Bifidobacterium adolescentis. Whereas some of the other, so we can have these, one way of making resistant starch has been to make these variations of cornstarch, which are called high amylose cornstarch. So essentially normally in plants, they make their starch with about 80%-ish amylopectin, 20% amylose, although it varies depending on the plant and even varieties of the plant. But you can find mutants of the starch synthesis pathways that'll end up making a more inverted relationship where it's more like 80% or 60, 70, 80% of the amylose and a lower percentage of the amylopectin. And it turns out that also makes it into a resistant starch. So those high amylose cornstarches, if you feed those to people that have both Bifidobacterium adolescentis and Rhuminococcus bromii, the Rhuminococcus bromii will win. It really loves those ones. And other sort of wheat and corn-based ones, whereas the Bifidobacterium adolescentis really likes the tuber-based ones. And you can see, interestingly, those two different species will now have different cross-feeding relationships with the other members of the gut microbiome. One of the interesting things about Rhuminococcus bromii is that it can't use glucose. Despite being a highly specialized starch-degrading organism, if you feed it pure glucose, it cannot grow. It needs to take in at least something as large as maltose, and then it can break it down to glucose internally. It still has the normal glycolysis pathways and all that. It just doesn't have a transporter to bring in the glucose in the first place.
Wild. That's wild.
Yeah, it's a pretty crazy one. But one of the consequences of that is that as it's digesting the resistant starch, there is some accumulation of that glucose around it that's now available to feed other bacteria. And the bacterium itself will naturally stick. It has binding proteins to stick it to the starch that's coming in. And other bacteria like Eubacterium recali, another bacteria that I've studied a lot, also does this and likes to co-localize with the Rhuminococcus bromii. So even though it can't break down the starch itself, it set itself up in a prime seat to eat that glucose and other smaller bits of starch that are being released by the Rhuminococcus bromii. And that Eubacterium recali is a butyrate-producing organism that can now take those residual monosaccharides, disaccharides, whatever is coming off there, and now produce butyrate. Whereas the Rhuminococcus bromii does not directly produce the butyrate itself.
I really love this because, I could make a joke and say sharing is caring. But the truth of the matter is, and I know right now in another room, Seth Bordenstein's ears are pricking up. The idea is that we are a collection of different organisms, and we look at each other in isolation, and that's not how we're living. And this business here, where for lack of a better term, please forgive this, the crumbs of what one organism is feeding upon is used by a different organism. And I love this concept taking place in us because we are innately narcissistic and we only really care about ourselves. Sorry, but it's true. But we're a series of ecological niches, and everything that we've learned about ecology takes place inside of us.
Yeah, exactly. And so, that's part of what makes this all complex to understand is that you can't just look at, is this organism present or not? Oh, okay, then we should feed you that polysaccharide, because that will feed that organism and will have good effects. It's the relationships between that one and other ones around it that are important too. And so if you had that Rhinococcus bromii and you fed it that high amylose cornstarch, but there wasn't those same bacteria that could surround it and then produce the butyrate, you're still not going to get the health benefits from it. If you look on the other side of it with Bifidobacterium adolescentis, it's not quite as giving of an organism. It doesn't, if you culture that one in pure culture, you won't see any carbohydrates, sugars accumulating as you break down resistant starch. It takes it all up itself. However, while it's fermenting, it doesn't produce butyrate, but it does release lactic acid as its main fermentation product. And there are bacteria that can take that lactic acid and convert it to butyrate. And so it's a similar concept, but instead of sharing some of the leftovers of the sugars, it's now taking its waste products and one man's trash is another man's treasure, so to speak. And it takes that and can convert that to butyrate. But again, so you can get a good outcome with feeding that potato starch to Bifidobacterium adolescentis as long as you also have those bacteria around to then convert it to the butyrate. So we got to understand all of those relationships. And there's going to be competitors coming in. So you mentioned the Bacteroides. They love degrading starch as well. And they're going to produce propionate instead of butyrate as their end fermentation product, which has slightly different health effects. And so probably we want to balance between all those things, but trying to control all that requires an understanding of those complex ecological relationships that are taking place there. And so if you back it up, and so you do a study where you feed 100 people a type of resistant starch, and you look at what happens to their butyrate levels, you'll see on average, across that population, the butyrate levels will increase. So good, it works. But if you zoom in on the individuals, you'll see maybe a quarter of those people did not have a butyrate increase and maybe even decreased their butyrate levels. And so one of the things we have to understand is what's happening in those cases. So there is those cases, as I mentioned, where there's people that don't have the bacteria that can break down resistant starch, but that's way less than 25%. There's other things going on there where there's different ecological networks that are present so that when a Bifidobacterium adolescentis breaks down the resistant starch, you don't get that lactic acid to butyrate production. Mm-hmm. Instead, you have other pathways taking place. And so understanding that, and if we can now understand those relationships, we can now predict based on someone's microbiota, which is going to be the better resistant starch, and for them to eat to get, if their goal is to increase butyrate.
It's
in some cases that might be the goal. Yeah.
What's interesting to me is something, and I don't know if this is related, but I would love to hear your thoughts on it. There's this, simplistic school of thought that there's a perfect microbiome. And, several people, and I'll put links in the show notes, have written there's no such thing as that. I think Ed Yong, for example, had a nice one in the New York Times some time ago. But the reason I bring this up is that, and this is critical for listeners and viewers, if you mentioned before, if you give 100 people this particular resistant starch, and they're going to get an increase in butyrate, but then when you zoom in and look at the individuals, you find that some didn't get that, and that has to do with their particular individual ecosystem.
Uh-huh.
And this is very similar to the idea of saying that 1 hectare of land in Brazil is just like the one a mile away, and it isn't.
Right.
And so what's important and what isn't, that core is the critical part. And I just simply love this idea because it's a really good way to show students, in my case, or the public in another, how sometimes these generalities don't really apply. And I really worry about overstating things in the sciences. And you've really done me a service here. Thank you.
Yeah, exactly. And it's not when you look at those 25%, it's not like necessarily they have an incorrect or wrong
Hmm.
microbiome, right? It's
No.
just that it's not necessarily compatible with that dietary component coming in and getting that particular outcome. And so it's a matter of we have to recognize those differences and find ways to account for it.
And how it works longitudinally.
Yeah,
Um,
exactly.
and an ecology is not static; it's always changing, and that's true inside of us and on our skin and in our armpits and other places. Absolutely, as much as in a Winogrodsky column, absolutely as much at the bottom of a pond or wherever it is you choose to look. The moving
Yeah,
finger.
exactly. And it's a really tricky question to get at because it's very difficult to do microbiome studies that longitudinally to follow it up. We've done things looking at the same person a couple of months apart and see a similar response to a resistant starch. So it was nice to even see that because you could worry that it's even day-to-day dynamics that could be
Hmm.
influencing that. But we on the scale of months, we've seen similar responses. But that doesn't mean on the scale of years that it would be, right?
Absolutely.
The other thing is with that, when you take that 25, we've done some of these experiments where if you take people in that sort of 25% that didn't respond to the one and feed them a different class of resistant starch, we can get butyrate increases
Hmm.
there. So suggesting it's that those differences between the resistant starches and differences between the microbiomes are both things that we kind of have to match up to get optimal results. And I'm sure that's true of many other fibers, probably true of drug treatments, probably true of all these other things that have these responders and non-responders. It's true of almost any intervention that you do with people.
You
People are
know,
messy.
well, and we don't really. This is going to be controversial. Please forgive me. I don't think we understand selection at the microbial level. I mean, we understand what we think is going on and what we intend. And a really quick example: when I took the Woods Hole microbial diversity course a million years ago, and I had no experience with anaerobes, but Abigail Salyers was teaching it, and so she had us. Please forgive me, isolate Bacteroides from ourselves. And you know
Mhm.
perfectly well how you do that.
Yep, I have done it.
Yeah. And anyway, in the name of science, right? There
Mhm.
you say it. But here's the interesting part. When I was a young person, I had pretty bad acne, and I was given a gram of tetracycline a day for about 5 years. I had to keep getting blood tests to make sure I wasn't killing the mitochondria in my bloodstream. I don't know, but it was something else. The reason I bring it up is that it had been over 20 years until I took this course. And there were still about 35% of the Bacteroides that I isolated from my own colon that were still resistant to tetracycline. And you would expect them not to be there.
Mm.
You would say there's no selection for it. Why is it still there? And this is something I'm very thoughtful about and makes me think that I'm not inferring intent, but we don't understand the perceptual level down there. And so I'm very thoughtful about it. And that's why I wasn't surprised by the idea of no perfect microbiome.
Yeah.
And what I found interesting about what you just got through saying is that even if you don't get, for example, the level of butyrate that you would like to see in an individual, there are other amendments or changes not involving the organisms you necessarily expect that can accomplish the same thing
Mhm.
because we're complicated.
Yeah, exactly. I talk about these different resistant starch degrading organisms, but even and I've talked about people that don't have them and that the resistant starch passes through.
Hmm.
There's some cases though where they don't have them, and we still get a butyrate response from the resistant starch. So we're still trying to understand what's going on there, whether there's just some other resistant starch being organisms we haven't recognized, or in some cases, communities can come together to
Yes.
break it down. So,
An old friend of mine, Bill Metcalf at the University of Illinois, used to say that the microbes have their own agenda and it's not yours.
Mm-hmm.
And it's really true, because when we study things, we want things to be true. It's very human, but microbes don't care about that.
No, not at all.
This is wonderful material here, and I think not only is it intrinsically interesting, I think it gives people a lot to think about regarding issues when they hear about increasing their fiber levels, which you recommend.
Oh,
Now,
yeah.
I presume that this means you don't suspect that there's some fiber supplement that would have the same kind of positive effect. I'm presuming you would think, and we've heard it elsewhere on this podcast again, just increasing certain cruciferous vegetables, that kind of thing in our diet.
Yeah, so the problem with going the supplement route is that it's going to be a given supplement is usually just like one type of fiber, and usually one type of soluble fiber particularly. And so what's going to happen is, so even when we do our resistant starch interventions and we feed people, we give them pretty large doses because we want to be able to see the effect that's happening. And we get this butyrate response. But the other thing we see happen is a decrease in diversity within the gut microbiota. Because you are now selecting for the ones that you're giving them a nice food source, and you're selecting for the ones that are going to be able to take advantage of that. And so if you don't have a variation in the types and amounts of fibers coming in, that's what you're going to do. You're going to end up selecting for a narrower set of organisms. And that is not probably what you want to do. There's all kinds of studies that say that diversity is one of the key things that you want to maintain within your gut microbiota.
It's a bloom that takes place, essentially.
Yeah, exactly. And the other part of it is, if it's all kind of easily accessible soluble fibers, that bloom will take place more early in the intestinal tract rather than later in the intestinal tract.
Right.
And there's been studies that show that actually we'd rather the opposite, but even better, probably an even amount throughout the whole intestinal tract.
I have to say, throughout your entire time here, I used to work in, I'm going to call it a biotech company, but it's not really the case. We were making bacterial polymers for food purposes. And we were trying to find the cheapest possible carbon source, and we were using corn syrup. And after I have been involved with signing for and receiving a trainload of corn syrup and seeing the differences from one lot to the other.
Yeah.
You have a very different view of these things. So I'm all for the FDA. Let's just put it that way. 100%. So this has been a wonderful, wonderful episode. If you had a couple of take-home lessons for listeners and viewers, the first is, of course, to increase your fiber content from fruits and vegetables.
For sure. Yeah, any whole fruits and whole vegetables, raw whenever possible, are going to be, your best sources of these. And you're also going to get all kinds of other beneficial nutrients at the same time. So that's definitely your best strategy. The other thing I'll just point out, though, is because everyone has this different microbiome, there's no one perfect microbiome. You might find some fiber sources don't actually agree all that well with you. That doesn't mean you should avoid fiber. It means you should try other things, other sources of fiber, to find the ones that kind of are most compatible with you.
I think that's really good advice. Darrell, many people today are into supplements for all kinds of purposes, and I know there are pros and cons, mostly cons. You made mention of several different genera or taxa of bacteria that might be very important for this process in the colon, of dealing with these recalcitrant polymers in particular. Is there any possibility of them being available as supplements, and would they be a good thing to take?
Yeah, so they're the ones that I talked about the most, Bifidobacterium adolescentis, Rhamnococcus bromii, as these main sort of resistant starch creators in the gut. Bifidobacterium adolescentis, along with other Bifidobacterium species, are actually commonly used as probiotics. So you can likely find some probiotic supplements of Bifidobacterium adolescentis. Ruminococcus bromii, on the other hand, it's not been used as a probiotic at this point. Not necessarily because it would be thought to have less health benefits, but rather because it's much more difficult to grow this organism. It's a very fastidious organism, and it's a very strict anaerobe. It does not actually grow very well on solid media plates. It does not grow very well unless you have very specific nutrient requirements in there. So it would be an expensive one to try and scale up and produce, and it wouldn't even store all that well either. Bifidobacterium adolescentis is a bit more hardy of an organism, so you can get away with doing that one. I also mentioned Clostridium butyricum there. You can find that one as a available supplement. It's not used quite so much in North America, but other places in the world it's actually used as a probiotic. The nice thing about that one is that it's a spore-forming organism, and so you have a very nice way of storing it long-term, even at room temperature, and not really having to worry about that. So then the question now becomes, do you want to take these as supplements? In terms of improving your resistant starch degradation capabilities, I'm not certain at this point that it's necessary, unless you were one of those rare people that I kind of mentioned that didn't have any resistant starch degrading bacteria in your microbiome naturally. There might be some benefits associated with that. But for most people, what's going to happen is you eat the resistant starch and you're going to naturally increase the levels of these things that are there already. So there's not necessarily going to be an added benefit to taking them as a supplement.
There was this interesting thought from microbial ecology in the 1930s by a person named Bas Becking that everything is everywhere and the environment selects. I'm not quite so sure that's 100% true, but it's absolutely true that we are a complex constellation of microbes everywhere. However, I'm wondering, just as precision medicine is making inroads by finding things that are kind of suited to the individual in terms of treatment, this might be important to the microbiota as well. So fingers crossed, right?
Yeah, for sure. And it might be, as we understand these things better, everyone might have one of these species at the species level, but there might be different strains, for instance, that have better health properties. But I don't think we're there yet at making those types of decisions.
The FDA often gives suggested recommended amounts of fiber in the diet. And if you look on food items, sometimes you see somewhat cryptic descriptions of things. I had to laugh at one that says polydextrose because I think they mean amylose by that, but I'm not sure. Do you have a thought of what people should look like if they wanted to purchase things that are prepared foods that had fiber, other than getting sauerkraut?
Yeah, so in terms of supplementing a bit of, if you want to get a bit of your recommended daily dose of fiber from these types of sources, sure. There's going to be, usually these are going to be soluble sources of fiber that they can incorporate relatively easily into their recipes that they're using to make these products. If that's your only source of fiber, that's going to be a problem because it's not going to be beneficial to only have one source of fiber, selecting for only a very narrow set of bacteria. And, we want to have a wide variety of fibers, wide variety of soluble versus insoluble, microbially accessible versus inaccessible, all these different things. We want these combinations, and you're just not going to be able to get that unless you are getting these from whole food sources and a variety of different whole food sources.
I also have become thoughtful about changes in the microbiome. Big move afoot to move toward, you know how I feel about this term, low carb diets, whatever that's supposed to mean. What effect does that have on the gut microbiome as far as this goes?
Yeah, so it depends. Sometimes people take a low carb diet to mean that any carbohydrate must be eliminated out of there. And that's going to have some detriments to your gut microbiome because those fibers, the resistant starch, these other dietary fibers, those are all within that carbohydrate category. And so if you indiscriminately remove all of those out as well, now you're taking away a major food source of your gut microbes. Now, that doesn't necessarily mean that they are just going to go extinct and go away, because there's other food sources available for them. Your intestinal tract, for instance, is coated with a mucus layer that is a combination, essentially, of protein and carbohydrates to make that mucus, and they'll start eating that. If they eat that a little bit, that's okay. That's part of the reason why we produce it. If they start overeating it and removing it, now we can have a lot more problems where we start getting inflammation in our intestinal layer, leaky gut syndrome, things leaking into the bloodstream, perhaps invasion of the microbes themselves across that layer. So lots of bad things that are associated with overeating of that food source, which is what they're going to do if you don't give them their preferred food source, which is the fiber.
That's what I was thinking. Once again, such a complex community.
For sure.
The other thing that I like to ask people on the podcast, if you don't mind, is your path to the microbial sciences. First off, I have to ask, and I apologize, do I hear Canada in your voice?
You absolutely do. So I am from Canada, Ontario, sort of a rural area halfway between Toronto and Ottawa. But I did my undergrad and PhD in microbiology at the University of Guelph.
Yes.
And then, so my PhD with Anthony Clark there. So I was doing cellulose research, so bacterial cellulose degradation, but his lab is actually much better known for bacterial peptidoglycan research. But he had started out earlier in his career doing cellulose and xylan research, and so it was sort of a holdover project. So I started out doing that kind of thing, and then I sort of switched teams over to Starch during a postdoc. Sometimes in the cellulose and starch world, it's viewed that way as kind of like different teams, but it's all bacterial polysaccharide digestion. And went over to Denmark, Technical University of Denmark, working with Birte Svensson. And she had done a lot of work with barley alpha amylases and glucoamylases, and really got me into these starch degrading enzymes. I came back over to the US, University of Michigan, to do a second postdoc with Nicole Kropotkin. And that's what really got me more into the gut microbiome aspect of it. And then when you're talking about starch in the gut microbiome, it's resistant starch is the main thing that's the focus. And so there I was lucky enough to be able to start my own lab at Penn State in 2017, when I've been here ever since. So,
one of the things I really like, and again, I want to emphasize this. Many people come to the microbial sciences with preconceptions. I see it in students all the time. And so when you think, hey, I'm working in food sciences, they'll think different things. They'll think I'm looking at the rumen. And in fact, I've had students get their PhDs in that area. Certainly, it's a great field. Or food safety. And we've had people talk about that. What I like here is this is really great, solid microbiology that at the same time has relevance to human health, but not in terms of a disease-causing organism.
Right.
And it good to know, right?
Yeah, and exactly. And, that's always been my focus, is on microbiology to help people in these sorts of ways. If you look way back, the reason I wanted to get into cellulose digestion by bacteria was when I came into high school, I was originally thinking, I think I want to be a lawyer. So imagine that, going from thinking I wanted to be a lawyer to now being a microbiologist. And what really converted me was this idea that you could genetically engineer these microbes to do all these amazing things. And that just really captured my imagination. And so, yeah, that's what really drove me into the microbial field.
I got to tell you, those initial experiences make all the difference.
Hmm.
And if I may be allowed this editorializing, I wish people understood the value in secondary schools of this kind of thing, just to get people thinking. And again, there are lots of people who make those efforts, and I have one coming on the podcast later in the year. But for the most part, and it's no one's fault, it's just the number of students and the workload. It's very much, here are a series of things you jump over, and you lose the joy.
Mm.
And you lose the opportunity that you, as a student, can find the things that really spark it in you.
Yeah.
I know that I can tell right now that you're inspiring lots of people from the way that you explain what it is you do.
Well, thank you. I hope so.
Yeah. So I want to thank you for this opportunity. I've really been looking forward to it, and I think there's a lot here of interest to a wide variety of people. And I want to wish you and your family and your research group the very best of luck.
Thank you. It was a real fun time. Thank you.
Thanks again. This has been Matters Microbial, a weekly podcast about the wonders of our microbial world and the people who study it. You can send questions, suggestions, or comments to me at mattersmicrobial@gmail.com. Show notes from today's episode, with epic links as always, can be found at microbe.tv/mm. If you like our work, please consider supporting us at microbe.tv/contribute. I'm Doc Martin, and you can find me in the biology department of the University of Puget Sound in Tacoma, Washington. Dr. Darrell Cockburn is Associate Professor of Food Science at Pennsylvania State University. Many thanks, as always, to David Renata for superb editing, and Reber Clark for the wonderfully quirky music. I hope that you've all enjoyed being part of our Quality Quorum for today. See you next time on Matters Microbial.